Our muscles move our bodies through their contracting action, pulling on our bones. But what makes this happen? Let's look closer. Whole muscles are made up of fascicles, or muscle bundles. Fascicles contain muscle fibers, which are long, multinucleated cells which are formed when many myoblasts, or pre-muscle cells, fuse together. Each muscle fiber contains hundreds of myofibrils. If we look closely at myofibrils, we can see a repetitive banding pattern which gives striated muscle its name. These bands are formed by overlapping strands of the proteins actin and myosin, also known as thin filaments and thick filaments. The Z-line marks the ends of single contractile units called sarcomeres. The I-band contains only actin filaments. The A-band marks the length of myosin fibers, which partially overlap with actin fibers. The H-band marks where the actin overlap ends and contains only myosin fibers. When the muscle contracts, actin is pulled towards the center of the sarcomere, so the H-band and I-band nearly vanish. When we zoom in even closer to the molecular level, we can see the molecular mechanics which allow myosin to pull on actin filaments. Each myosin protein has two protruding heads with actin binding sites. Adenosine triphosphate, or ATP, drives the cycle of conformational change that the myosin heads go through to pull actin fibers. Initially, an ATP molecule binds to an actin-bound myosin head, releasing the actin-myosin connection. The ATP splits into adenosine diphosphate, or ADP, and a phosphate ion, and the myosin head reattaches farther down the actin filament. The phosphate ion is then released, causing the myosin arm to swing back, pulling the actin filament towards the center of the sarcomere. The ADP releases and the cycle begins again.